1. Observing root hairs in germinating seeds. Soak some gram seeds in water overnight. Take a petri dish or plate and place wet cotton wool on it. Arrange the soaked seeds on the cotton wool. Keep the setup in room temperature for a few days. You will see that white outgrowths come out of the soaked seeds. This is the radicle which forms the root system of the plant. Observe the radicle under a microscope. Tiny root hairs can be seen to show that light is necessary for photosynthesis. Take a healthy potted plant and keep it in a dark room for 24 to 48 hours. To de-starch its leaves, make the leaves starch-free. Select any one leaf and fix a strip of black paper on the middle of both sides with the help of clips. Expose the plant to sunlight for 6 to 8 hours. Now plug the selected leaf. Remove the strips of black paper and perform starch test. It will be found that only the covered parts of the leaf give a negative result, that is, turn brown with iodine, while the other portions of the leaf turn blue-black. Any other leaf of the plant will also turn blue-black with iodine. This shows that photosynthesis occurs only in those parts of the leaf that receive light. 3. To demonstrate that chlorophyll is necessary for photosynthesis. Take a healthy potted plant having variegated leaves. A variegated leaf is one which has both green and non-green portions. Example, the leaves of croton or coleus. Keep the plant in a dark room to de-starch the leaves. Select a leaf and mark its green and non-green portions. Keep the plant in sunlight for 6 to 8 hours. Next, plug the selected leaf and carry out starch test. It will be seen that the green part turns blue-black with iodine, while the non-green portions turn brown. This shows that photosynthesis occurs only in the chlorophyll-containing parts of the leaf. To demonstrate that carbon dioxide is produced during respiration by germinating seeds. Take a flat bottom flask and place wet cotton wool at the bottom of it. Take some soaked seeds and place it on the cotton wool. Close the mouth of the flask tightly with an airtight cork. Keep the setup in normal light and temperature. After a few days, the seeds germinate. Now open the cork and tilt the flask over a test tube containing lime water. It is seen that the lime water turns milky. This shows that carbon dioxide is given out by the germinating seeds during the process of respiration. 5. To demonstrate anaerobic respiration in germinating seeds. Take some germinating bean seeds and remove their seed coats. Take a test tube and completely fill it with mercury. Introduce the soaked seeds into the mouth of the test tube. Invert the test tube on a trough containing mercury. The seeds will reach the top of the test tube. Keep the setup undisturbed for 1 to 2 days. It will be seen that the level of mercury in the test tube comes down. Introduce a pellet of potassium hydroxide into the test tube. The level of mercury again rises in the test tube. This shows that the gas which collects above the mercury level is carbon dioxide. This experiment should be demonstrated by the teacher as mercury is highly poisonous and should not be handled by children.